So first thing is thanking the organizers, Kieran and Dario and Alice, for inviting me again to this beautiful events that you are organizing within the HybridNeuro project. So actually, it's very nice to be surrounded by people who know a lot of the things I work on and probably better than me. So that way I can learn a bit while I speak aloud. And it's also true what Dario was just saying, because we had no discussions whatsoever. I was fearing that because Dario was going to be speaking before me, maybe there was going to be a bit of overlap and I had to figure out what he was going to be talking about. I more or less managed to be right in that. So well done, Dario. Good. So this is going to be a much shorter presentation, which is also good for everyone. So let's have a chat on what we think is going on with regards to what's coming to the motor neurons and how we use it. Before doing that, let me very briefly introduce you to my current place of work. As Dario was saying, I moved a couple of years ago back to my hometown in Zaragoza, and I just add this slide here. Oh, by the way, should I minimize this? And that way it doesn't... Yeah. It's better, right? Yep. Okay. So just in case at some point in your lives you decide to pass by, you can come to our labs. We will show you how things are done then. We can stimulate your brains, your muscles. We have everything we need, okay? We have needles as well. You're invited. And you can also maybe have some fun around. It's a beautiful place, close to the mountains, yeah, a lot of stuff. And there I work in a group which is a very nice group to work in, because they don't do anything related to what I do, but they know a lot of stuff with regards to signal processing. And actually, many times you see very nice synergies there. And actually, funny story is that Dario knew the professor there who built this group years ago, just because he took some of the ideas in their research in cardiac signals and implemented them. We will have a quick chat on that in a couple of slides, but it's a nice, smooth path, right? So these are the kind of research lines that we are covering in our group there. And as you can imagine, I do just a tiny little bit with regards to neural interfacing. The other things are done by others in this large group. Funny stories in Spain, we have groups that are massive with lots of things being done within them. Okay, so this is a summer school on the topic of human motor augmentation, right? If I'm not wrong, I hope I'm not. And that means we're trying to figure out ways in which we can expand the capabilities that humans have to interact with the environment. And one of the key elements that one has to take into account when doing so is trying to figure out ways. So it's the neural resource allocation problem, right? How do we figure out how to extract meaningful information that we can use to control expanded degrees of freedom, right? And the first thing that comes to mind when thinking about this kind of problem is that we have to pick up these signals, and probably the best would be, or ideally we would like to pick them up from our brains, right? So if we can selectively record from specific areas or neural populations and use them to map their activity on some specific behaviors of external devices, that way we could test this possibility of expanding our human capabilities. But there is a challenge in recording brain activity, as probably all of us know, because if we want to go for invasive methodologies to record specific activities of neurons or populations of neurons, that's going to involve pretty invasive approaches that at present are still quite challenging, technically demanding, etc., expensive, and above all, they have some associated risks. So you are not going to have a justification to use them in most cases, right? Only in very specific cases with high levels of disability. On the other side, if we try to record activity from the surface of the brain, trying to pick up some brain activity using scalp electrodes or non-invasive methods, we're always going to be facing this challenging problem of solving the inverse problem of relating field potentials with neural activity. And this is always going to be very challenging, because there are going to be, we don't even know how many sources are we going to be looking at or staring at when we are recording these kind of field potentials, and how many sources are being summed to generate the kind of activity we pick up from our sensors, right? So it's a very challenging problem here. Okay, so then going back to, there are going to be, I think I counted them, maybe 3 slides that are slightly similar to Dario's. This is one of them, because this is the kind of idea he was discussing before and repeating now a bit. So if instead of thinking about picking up signals from the brain directly, we try to think about how possible would it be to record them from the other side of this neural network, we obviously start thinking about motor neurons, which are the output layer of our nervous system, right? So these are useful sets of neurons because they are accessible, as Larry was mentioning before, and also because they have very important connections with many different parts of the nervous system. So in theory, if we can record from them and separate somehow the kind of activity that is driving their behavior, then we stand a chance of figuring out activity that is coming from elsewhere in this nervous system, right? And I will, so this would be the kind of question that we are going to be asking ourselves in this presentation. Are we able to record activity from motor neurons that provides us with some meaningful information about ongoing activity in the central nervous system? And can we potentially use it for different applications, among which for this event, human motor augmentation would stand out, right? So what would be the advantages of this kind of interfaces? This is repeating a bit what Dario said before, but in a sense, when we are recording from muscles instead of recording neural activity per se, we have several advantages. The most obvious ones are going to be robustness, the fact that it's an unobtrusive technology because we don't have to go very invasive to collect these kind of signals, or the fact that we can deploy or use wearable technologies that are out there right now. Many groups and companies are developing these kind of technologies these days and are allowing us to record in a much more wearable way this kind of information and process it with advanced tools that we are developing. And apart from these advantages, I add one more that I think is very useful, or I think it's one of the keys from a neural side of, from a neural perspective that motivates the use of this kind of technologies, which is the selectiveness. And what I mean by that is that it's kind of expressed or described here, and I will go back to it in a later slide. But basically, what I mean by that is when we were discussing before the challenges that we would address when we try to record from brain activity directly is the fact that many sources are going to sum up and are going to be mixed, and we don't know how to separate them. There's no solution for that. Each of us is going to be completely different, and the brain is going to be active in many different ways. So it's an unsolvable problem. We will never have enough spatial resolution from the outside. Now, what happens is that if we look at the muscles and ideally the kind of activity that is happening in specific areas, especially from the motor cortex, but potentially from other places, and this kind of activity that is selectively happening in a specific circuit is transmitted throughout the cortical motor system to the periphery and the muscles, then if this information can be detected and decoded reliably from the muscles, that means that we are effectively using our nervous system as a special filter that allows us to separate that source from others that could be mixed with when we were recording them from the brain. So that would be, I think, an important reason why we could explore these technologies, right? Because we are kind of enhancing massively our spatial resolution of what is happening in selective, functionally selective circuits. Okay. So that is the kind of idea that we are trying to explore here, whether we are able to look at and separate these neural components that are driving motor neuron activity and motor units and try to extract them, separate them from the actual motor commands that drive our behaviors and try to use them for this kind of enhanced applications or to infer certain aspects of neural activity in the central nervous system. That would be the overall framework. Now, if we want to do that, or in order to assess whether this is possible, we need to check how signals are going to be transmitted and converted on their way from the central nervous system, supraspinal regions in the brain, and down to the muscles. And I identify here with these three boxes, 3 parts of this transmission system that are going to play a very important role in how the signal gets transformed. down to the way where we are picking up the signal, right? So if we separate them, we have these kind of three important steps. And if we go from the end back to the origin, we could identify this first step of going from the EMG to motor neuron activity. Then we would have the next step, which would be going from the motor neuron activity to the common inputs that neurons are receiving. And then the next step would be going from these common inputs to activity that is taking place in the nervous system in the brain, for example, right? So these are three steps that we have to take in order to be able to properly say that whatever we are picking up from our muscles is relevant with regards to whatever's going on in the central nervous system. So let's go one step at a time, shall we? Of course, the first step would be assessing whether or not we can decompose motor neuron activity from interference electromyography signals. And of course, I'm not going to spend much time here because Dario has covered much of what one could potentially say about this. And the whole point is that at present we have advanced quite a bit, and we have already technologies that are minimally invasive or non-invasive that allow us to record from hundreds of channels from many different muscles and decompose this activity under certain restrictions and allow us to address the problem of identifying motor neuron activity from a population perspective, which is very handy because many of the things that we can extract in terms of neural information is going to be encoded or best decoded from populations instead of single units. Right? I would say that if we were to identify challenges at this point with regards to this step going from interference signals into motor neurons, this would be the main challenges that I could think of at present and that we need to keep working on in the future. And hopefully many of the things that Dario covered before end up giving us solutions that allow us to solve them, especially the fact that we depend on the kind of muscles we are recording from. All of the people here who have some experience with surface electromyography know that things can change quite massively across subjects and across muscles. So this is an important element to take into account. Then how much things change when in more challenging conditions beyond what we typically do in the labs, which is isometric contractions. And then how we can implement actually technologies that are properly useful in unconstrained conditions where recordings can take as long as needed, etc., which regards to the development of new and more wearable kind of sensors and electronics. But anyways, this would be the first step. And then we go to the next one, which would be trying to figure out what kind of neural inputs motor neurons receive based on the activity they are producing. That would be our next step in our identification of whatever's happening at the beginning. As discussed before, motor neurons, as individual cells, they behave in very nonlinear ways, right? We can use models like the integrated pulse frequency modulation model to characterize how these individual cells behave in the context of or when they are receiving different types of inputs. This is a very largely used model, as I was saying before, for example, used to characterize modulation, so how the autonomic nervous system modulates the activity of the ECG. And this has been extended then to characterize activity of motor neurons, and it has been quite extensively explored and characterized in terms of how the spectral contents that are at the entrance of motor neurons represent the output. So when we characterize motor neurons using this kind of models, which is a very similar model to what would happen in reality, and then we simulate different sets of inputs at different frequencies, covering a range of all kinds of frequencies that one can think of, then we should expect to have at the output in terms of spectral contents of the neuron that is generating this post or this point processes or pulse activity that has an expression like this one before, which this expression here, which is of course very messy, but we can identify different parts in it, right? So one of the parts that this expression has, this is the power spectrum of the activity at the output of a motor neuron when the input is this summed activity of sinusoids. right? So what we can identify here are these three parts. One is the DC component, is the net activity that is driving the motor neuron, and because this is a positive type of signal, we're always going to have this kind of DC component, and this is the yellow or orange box over here. Then we have this green part that relates to the actual inputs that the motor neuron is receiving at the different frequencies. You can see the delta functions in the frequency domain at the different located at the different frequencies of the inputs, right? At F1, F2, etc. And then we would have this red part here, which would relate to the nonlinear parts components of the neuron, right? If we represent that using a Fourier spectrum, that relates to these graphs over here. These are for different simulations where the modulating frequency is changed between 0.1 and 0.2 in three graphs, and we can identify the DC component. Then the green part is the modulating signal that we would ideally like to identify, and then there's all these nonlinear components that are driving activity. Now, it's important to note here that these kind of derivations, mathematical derivations, as I was saying before, they've been conducted for other kinds of purposes, like cardiac signals, where the kind of modulators that are driving activity are in very low frequencies. So if the heart is beating at, let's say, 1 pulse per second or so, then the kind of modulating activity that we want to investigate is happening at much lower frequencies. That is the reason why we see it over here without overlaps with the nonlinear components in the output of the motor neuron, and that would effectively mean that if we use a low-pass filter, we could potentially extract this information more or less, right? But in our problem here, trying to derivate the activity in the central nervous system or in the brain from motor neuron activity, we are extending a problem where we likely are going to have frequency components that are going to span a much broader frequency band, right? And going beyond the actual discharge rate of the motor neurons, which would be this peak over here, right? So potentially this kind of activity would not be solvable or decomposable in this kind of domain, right? Now we can see graphically this here, and this is a simulation where we have a set of motor neurons. In this case, it's a simulation with 177 motor neurons. And what we've done is we have simulated several common inputs at different frequency bands, and we have simulated these motor neurons all receiving this kind of common inputs and then receiving sets of independent inputs, each of them. And then we have obtain the power spectral density of this of each of these individual motor neurons. Is what you can see here, as in line with what we showed before in the previous expression, is that the spectrum is dominated by the nonlinear components of the motor neuron, right? And which is in line with what we showed before. So if we see this, we can clearly see that if we took individual cells and try to trace back what kind of activity is driving them, it would be a challenging problem because we would be having a spectrum that is massively dominated by the nonlinear components of the cells themselves. Now, what's useful, and this is beautiful work that Dario did already 10 more years ago, is that when we look at the population activity, when we start putting together more and more motor neurons, and of course we can do that now that we can decompose, but we can also simulate that. And then the analytical derivation of this kind of combination of motor neurons, having the activity of tens or hundreds of motor neurons, is something that is shown here, where if we relate it to the previous case where we had an individual cell, we're going to have 4 highlighted boxes. One is the DC component of the summed activity, it's still a positive process. And then we have this activity in green, which still relates to the actual inputs that the motor neurons are receiving. And because all of the neurons are receiving this information as a common input, their activity is going to be summed up coherently, and that's going to mean that it's going to be multiplied by a factor that increases as a function of number of neurons that we're using. And then here we have two more elements in this equation, the nonlinear components that we were describing before, and the summed activity of the independent inputs that each of the neurons are receiving. But because none of these components are going to be summed in a coherent way, in the end, as soon as you start increasing the number of neurons that you're taking into account, the way of these components into the activity, into the summed activity that you are looking at when combining neurons, is going to be reduced relative to the common inputs. So this would imply, in theory, that when we have access to large enough amounts of motor neurons, And when we sum their activity, what we should end up having is a very neat and clear reflection of the common inputs that these neurons are receiving, okay? So this would be a graphical representation of what I just said. If you look at this, then when we have few units, then their spectrum is going to be dominated by the nonlinear components that they produce when firing at specific rates. And as soon as you start increasing the number of neurons combined, then these kind of nonlinear parts are going to be canceled out, and we're going to be left with the linear part related to it. So in graphical terms, we can see this again. So this is the same graph as before, where we were seeing individual cells. And when we do the power spectral density of them, we see that they are dominated by the nonlinear components of the motor neurons. And as soon as we start summing their activity of large enough sets of motor neuron populations, we are starting to have this kind of graphs like the one over here, where we can see that the peaks of the power spectral density are directly related to the common inputs that the motor neurons are receiving. Okay, so if we had this kind of activity, then it would be trivial to say that if we have adequate bandpass filters, we're going to be able to extract the information that is driving the population of activity, and that way we can infer certain kinds of activities, and importantly at any given frequency, right? Because so long you have a large enough set of neurons sampling the same process, that would mean that we could sample even very high frequencies of information that reach the motor neurons. Okay. In more graphical terms, you would see that we could have, if we have these kinds of inputs at the entrance of the motor neuron pool that are the common inputs that are driving their activity, then if we extract if we sum the activity of all the motor neurons that we have access to, and then we bandpass filter them in different bandwidths, we would end up having signals that relate massively with the input. This is obviously in very ideal cases where we have a very large amount of motor neurons receiving exactly the same kind of information. And this is useful, as I was saying before, because you can combine, as said, this applies not only to very low frequencies that would be related to how we move, these kind of frequencies that go between 0 and 10, 12 hertz, and that dictate how the muscles are moving, but also this also applies to higher frequencies that can be related to other activity that takes place in the central nervous system, like beta, gamma rhythms, etc. Right? So ideally, this kind of information is also going to be sampled very effectively by the population, by the neural population, by the population of motor neurons. And this is interesting because this kind of activity, even though it's going to be sampled and transmitted by the motor neurons behaving as linear amplifiers, as I was saying before, this kind of information won't have any effects, any actual effects on how we move, because due to the biomechanics of the muscles that are receiving this kind of information, higher frequencies are not going to have a massive impact on movements, actually non-impact at all, as has been described in different papers in the literature, especially this kind of old papers written by Baldissera and others, who show a very clear difference between neural transmission of information and how this then relates to produced force by muscle fibers. Okay. So this is kind of a summary of this part where we say that if we have large enough sets of motor neurons, in principle, we should be able to trace back whatever is going on at the entrance of these motor neuron pools. And so in theory, that would imply that we are able to extract this information. Now, a bit of a break there. One of the things that can pick our attention in this regard is what I was showing you now was all based on simulations, and these simulations are kind of ideal because they are just simulating kind of noise inputs that are entering into all motor neurons as independent inputs, and then we have some common signals that are simulated in different bandwidths, and we can see that at the output we can trace them back, and we have spectra like this. And this is ideal spectra that shows or highlights very neatly the common inputs they are receiving. Now, if we look at actual power spectra of experimental data that we obtain in human subjects, this doesn't look exactly like that. What happens is that we have a spectra that are still dominated by the nonlinear components that individual motor neurons typically present when we look at them individually. So how can we explain that, right? This blue line here is the average of, I don't remember now how many, but probably 15 subjects where we have been able to decompose relatively large sets of motor neurons for each of them. We have produced the composite spectrums for each of them, generated the power spectral density, and plotting them here. And we have this kind of spectra here where we have a dominant peak, very clear dominant peak at 10 hertz, which is pretty tightly related in this case with the discharge rate properties of the motor neurons that we have decomposed, because in this case what we are doing is mild contractions at around 10% of the maximum voluntary contraction. So this kind of contradicts what we are having here. Instead of having something as we would anticipate, which is an enhancement of whatever common inputs that motor neurons are receiving at different frequencies, we still have a dominance at this kind of discharge rate related frequencies. And there's no explanation, clear explanation to that, right? So one of the things that we've been questioning lately in our work is whether our assumption regarding the kind of inputs that motor neurons are receiving is correct. So what I mean by that is that in the previous slides, what I was showing you, both in analytical derivations, simulations, etc., was based on the assumption that any kind of common inputs that motor neurons are receiving are continuous processes that are somewhat stationary, wide-sense stationary, and that means that they kind of slowly vary across time, and they have some spectral information in contained bandwidths. And this is rightfully so to assume something like that, because when we think about movement, we think about a continuous process, right? So why on earth would we be receiving something that is not like a continuous progressive process? However, that doesn't have to be necessarily true, because we know from many different studies in motor control and also in other aspects of motor neuroscience is that many times processes happen in very brief events or bursts. So one of the things that we were wondering and trying to address in our research is whether, instead of assuming that everything that motor neurons receives is a continuous slow changing process, whether we should expect to have also impulsive type of signals that are driving motor neuron activity. Impulsive, this is related in signal processing theory, is related to impulsive noise, which is a kind of noise that is a very particular one because it occurs only randomly and at seldom times, and it produces a very large amplitude kind of signals that change the characteristics of the signals at those points in time, but the rest of the time the signal is still producing a more stereotype kind of activity that can be modeled with autoregressive models or whatever. So then the question is, how can we check whether motor neurons are receiving this kind of impulsive signals that every so often disrupt their activity, so to speak, right? Well, before addressing that, we can think about simple experiments or different types of evidence that suggest that indeed we should expect actually to have impulsive inputs in the motor neuron pool, actually, right? So, for example, these are very old papers by Mial and others, and there's been quite a long trace of research in this regard, discussing about intermittent control of movement, where the authors were describing how by recording the force traces you could see, depending on the characteristics of the feedback that they were providing to subjects, that sometimes forces produce very sharp changes, and they even tried to describe at which rate this kind of disruptive activity was taking place, which was something between 0.5, 0.8, 1.8 hertz. This could be potentially an example of kind of impulsive signals, right? Some sudden changes in force that then goes back to smoother type of evolution. This is another example. This comes from a group in Italy who is doing very beautiful research, and in this case, what they were doing is asking subjects to hold sustained contractions, and every so often, without it being predictable, the subjects received unexpected stimuli, either electrical or auditory stimuli, and this caused very salient responses at the brain level. And then they looked at the force traces and saw that the force was showing a very brief as well reflection in force that was tightly related with whatever was going on in the brain. And all these kinds of things are happening actually in very brief amounts of time. So that means that it's not like the motor neurons are changing massively their discharge rate. They are just changing for a very brief amount of time their behavior. And finally, of course, and we will discuss this later, this relates to Mario's work that Dario was mentioning before. We also have examples in the literature when describing oscillatory activities, like, for example, beta bursts that occur in the brain that are very brief periods of time where oscillations are occurring in the brain, and this kind of activity is also transmitted to the muscles and can produce disruptions in the muscle activity. All of these can be thought of as examples of impulsive type of inputs that all of a sudden disrupt activity of motor neurons. And if we simulate this kind of activity, what we are expecting to have is something like what you see on the bottom right corner of the slide, where you see that at a certain point in time, if this is true, these kind of impulsive signals are going to be able to transitorily synchronize all the motor neurons, because all of a sudden they are receiving an input that is common to all of them and is very, very brief in time, but also very sharp. So this is going to cause that at some point in time, all the neurons behave as if they were a single neuron. And if you think about the power spectral density that I showed you before of experimental data, where all of a sudden we start having again this kind of nonlinear components in the population, this would be a way to see this from an experiment, from a simulated perspective. If we all of a sudden synchronize all the neurons, what we are going to have is a behavior that is becoming less desynchronized and more like a single neuron process, right? So this is the kind of question that we are thinking of right now. And then the question would be, how can we identify these events if they actually exist experimentally? How can we spot them when we are recording motor neuron activity experimentally? So one of the ways we could do that would be to find these moments where all of a sudden motor neuron pool is showing. When you sum the activity of the motor neuron pool, you all of a sudden see very high levels of activity at the frequencies associated with the discharge rate, because this is the kind of activity that we were seeing before in the power spectrum that was dominated. So this is what we did. We did that in experimental data and also in simulations, considering different types of inputs received by motor neurons. This is what you have here. We have experimental data, and we also have some simulations that either consider impulsive common inputs to the motor neuron pool or continuous processes like oscillatory activities or non-input whatsoever. And what we could observe when looking at these graphs over here, what they are showing you is the average of a measurement that characterizes, let's say, the synchronization among motor neurons on a population level is called spike distance measure, and it goes down as soon as the motor neurons start being very synchronized. So when all neurons are firing at the same time, we would have a measure that would go down to 0, and whatever goes in between. So when we find those time points during our processes where the discharge rate power increases suddenly, and we then average whatever's happening with regards to the spike distance as measured here, what we can see is that experimentally we see a very clear dip in this spike distance metric, which is kind of showing you that during these periods of time where the power at that frequency increases, there is a very high level of synchronization across all the motor neurons that are being recorded from. When you do the exact same thing in simulations with matched numbers of motor neurons, and you simulate bursts of activity that every so often occur at a rate of, let's say, 0.5 or 1 hertz, for example, and they are able to depolarize a number of motor neurons, then you can see that similar behaviors are obtained in this kind of framework. However, when you don't simulate impulsive inputs, but instead you simulate continuous processes or non-inputs at all, you see things that are pretty milder and are pretty much related to chance levels of motor neurons happening all of a sudden to fire at the same time. But you can see when you run a statistical test on this kind of data, you can see very clear significant differences between these kind of conditions, both experimental and bursting activity, bursts or impulsive signals and other continuous processes. When we then look at the power spectral density of these kind of processes, maybe one would predict that when you are simulating this kind of impulsive inputs to motor neuron pools, that the spectrum would behave in a very different way to what we actually see experimentally. Because if you, for those of you who are familiar with Fourier transforms and signal processing, if you do the Fourier transform of a train of delta functions, you would expect to have a train of delta functions as well, which would be the effective input that the motor neurons are receiving. So maybe we would have that kind of input enhanced in the power spectrum at the output. What we actually have is this kind of behavior, which is very similar. Again, the y-axis is not normalized across these conditions, but again, we have a spectrum that is dominated by the discharge rate frequency of the motor neuron pool and their harmonics. And in terms of shape, it kind of resembles quite neatly what happens at the experimental level. On the contrary, when you simulate other kinds of continuous processes, you typically have a peak at the frequency of these continuous processes that you're simulating. And moreover, when you look at then a typical measure that we use when analyzing motor neuron activity, which is the intramuscular coherence, which characterizes the kind of common inputs from a spectral point of view that motor neurons are receiving, we see that unlike in the continuous inputs scenario where we typically see peaks at the frequencies of these simulated common inputs, what we have when we simulate common inputs in the form of impulsive processes is something that is associated with a very clear trough at the frequencies of the discharge rate and harmonics, meaning that we are actually abolishing this kind of common activity across the motor neurons when we are inputting the kind of impulsive inputs that we are simulating in these cases. What we are doing is we are kind of resetting the phases of all the motor neurons, and then they go back into firing at their own times. And this causes that the intramuscular coherence goes down at these frequencies, which is pretty much in line with what we see experimentally. There's a bit of explanation to this kind of metrics, but what's nice here is that by using impulsive inputs, we can replicate very neatly what happens experimentally in the conditions where subjects are performing sustained contractions. And so this is kind of looks to us as a very strong argument to propose that we should start thinking about different types of inputs, not just continuous oscillatory processes, but farther from that, this kind of impulsive inputs. And if we think about this kind of inputs, then we should be thinking what is the impact of this kind of activity in terms of the behavior of the motor neuron pool. And going back to the main question in this presentation here, how well or how capable are we going to be to decompose the activity that motor neurons receive based on their outputs, right? So one of the things that happens, or the consequences of considering impulsive common inputs as something that takes place at the motor neuron level, is that the motor neuron pool is going to start behaving less and less as a linear amplifier, as we were describing before, and as we would expect in principle. And this is because as soon as the processes start getting synchronized every so often, our spectrum is going to be looking more and more like this kind of shape where the peak at the discharge rate of the population, the average discharge rate of the population is going to have a dominance in the spectrum, this and the harmonics. And so this is kind of showing a very clear nonlinear behavior at the motor neuron level. And if we then think about this is another way of seeing this nonlinearity at the motor neuron pool. We can simulate processes that are either impulsive inputs, trains of impulsive inputs, and then we can compare that to a continuous process that matches in terms of power at a given frequency, and we can see that the outputs are going to be different, which is a very clear way of showing that the system is not behaving in a linear way. And this has an impact, of course, when we want to think about how we can extract information. So if we then simulate a framework where we have motor neurons receiving common inputs that are a sum of impulsive types of inputs and then continuous inputs, the continuous inputs are going to be decoded or extracted less reliably from the motor neuron activity, just because the impulsive inputs are going to be pushing the system into a nonlinear behavior, right? So this is a limitation in terms of how well or how capable we are going to be in terms of decoding the activity at the outputs, at the inputs based on the outputs. So here, what I'm showing you here are three examples or three cases where we either don't have any impulsive inputs simulated in our system. This would be the blue line here. And this is for two scenarios where we either have 10 neurons or 177 neurons. And you can see as soon as we start having more and more neurons, we're going to have a nearly perfect decoding of the input based on the outputs. However, as soon as we start simulating impulsive inputs, this decoding is going to be massively affected and it's going to go down. And what's worse, it doesn't matter how many neurons we have access to, because of course these impulsive inputs are going to distort the activity of all these neurons at the same time. It's going to cause that their sampling, their independent sampling of the continuous processes they have at the entrance is no longer done in a linear way, so to speak. Okay. So this is just to wrap up the first part of the presentation. I think I'm more or less fine. Just to wrap it up, we can say that we can have reliable estimates of the inputs at the motor neuron pool level based on the outputs so long continuous processes are simulated. But as soon as we start assuming that other kinds of processes are also driving the motor neuron activity, we should anticipate that maybe things are not so straightforward, and we should try to push for better methods that take this kind of activities into account. Now I will go fast into the next part of the presentation, which relates to how activity gets transmitted between the cortical circuits that we are interested in decoding in our framework of looking at muscles and tracing back cerebral activity based on the activity that muscles receive. So this would be the last step that we were describing before. And I make this bit of a mess here in the connection between the brain and the muscle because so for long already it has been shown that pyramidal tract neurons and descending neurons that go from the cortex downstream to the spinal cord, and these kind of neural cells are very well entrained with ongoing activity in the motor cortex. So that means that effectively they are sampling the kind of processes that are taking place in the brain and sending them downstream to the muscles. And then it's also been described very thoroughly that these kind of processes then drive motor neuron activity in a way that when we look at the power spectrum, the spectral coherence between muscle activity and brain activity, we can see evidence that the information is being transmitted in a linear way between the brain and the muscles. However, in many previous papers in the literature, it's been described repeatedly that even though this transmission can happen in a linear way, because of the fact that the different actions that are transmitting this information are going to have or present very different properties in terms of conduction velocity in the transmission of information, what we should expect is that the kind of transmission that takes place across different subjects, tasks, and conditions is going to vary largely in terms of transmission delays and the characteristics of this transmission. This would impact our previous proposal of characterizing activity in the brain based on the muscle activity, because we would never have a stable enough system that would drive activity in a reliable way if that was the case. So we checked this kind of framework a while ago, and we tried to model this kind of distribution of conduction velocities that the axons in the corticospinal tract have and that connect the brain with the muscles by using different types of simulations where we were simulating different frequencies being transmitted by channels at different transmission velocities. And what we could observe in these kind of simulations is that in the end, if you can think of this as sets of upper motor neuron cells that group together with specific delays, they are going to behave as different channels transmitting different versions of the same signal with different delays, right? And so at the entrance of the motor neuron, what we should expect to have is a kind of like linear transformation of whatever's happening at the input, so long we assume that this kind of activity is transmitted uniformly throughout all these actions that are connecting brain with the spinal cord. So by doing that simulation, we could observe that what happens is actually that at the entrance of the motor neuron, what we should expect to have is the kind of activity that is happening at the brain level that is being transmitted by the fastest channels that are transmitting this information, streaming this information downstream to the motor neurons. And the reason why this happens is because there is a natural echo cancellation effect in this kind of systems. Whatever is happening at delays larger than the fastest route connecting the brain with the muscle is going to have a cancellation effect, because there will be replicas of that activity happening before and after that would naturally cancel this activity. So what this implies is that even though there are multiple channels that are connecting the brain with the muscles and transmitting the same information with many different delays, at the end, due to the characteristics of the distribution of the axon conduction velocities that we have in our systems, what we end up happening at the entrance of the motor neurons is a faithful representation of what's happening at the brain level, just with a delay that is dictated by the fastest routes connecting the brain with the muscles, which for upper and lower limbs is something around 8 to 10, 12 milliseconds. So this can be experimentally validated by looking at conduction velocities of MEPs, which are known to be related to the transmission velocity of the fastest axons connecting the brain with the motor neurons, and relating that to the transmission of other more ecological signals, like, for example, beta processes, right? So we've been doing quite a bit of research over the past years trying to validate these kind of ideas and showing that they are useful because they effectively mean that what we have at the entrance of motor neurons is very tightly related with ongoing activity at the brain level. This is work done by Mario Bracklin some years ago, where he would show that this kind of ongoing activities is at the muscle level at the specific bands. For example, here in the beta band, around 15 hertz, 15 to 30 hertz is pretty tightly related to ongoing activity at the brain level. And also because this kind of activity occurs in very brief periods of time, because of the fact that we have such stable transmission system, we are able to trace them back very neatly with our decoding methods based on muscle activity, right? Also recent work by Emmanuel, who is around here as well, he's showing that this kind of information is being transmitted to all motor neurons that are sampling this kind of processes, regardless their recruitment threshold. That means that this kind of activity reaches the motor neuron pool in a uniform way, which is very handy as well. And we've also been showing before that this kind of activity has size effects of changes that are comparable to whatever we have in when we're recording activity directly from the brain, which is very handy as well. Of course, as Dario was saying before, we can use this concept and use it to implement neurofeedback types of approaches where subjects are delivered or given information about what's going on at their muscle level, ask them to modulate this kind of activity. And in theory, if our assumptions work well, in the sense that this kind of muscle activity is representing directly whatever is going on at the brain level, what we would expect is that if subjects are able at changing specific aspects of ongoing oscillatory activity in the muscle level that we think is related to brain activity, then we should see an associated change or related change at the brain level in the same direction as what we are imposing or asking the subjects to drive at the muscle level. This is something that we were able to show to some extent in previous works by Mario as well, which are very interesting because in the end they show that there's a tight resemblance in the changes that are occurring at the brain and muscle level, and effectively they show that we can use muscles in a way to implement kind of brain-computer interfaces. And just to go back to these kind of concepts that I was showing you before, and I will skip one slide maybe for the sake of timing. This kind of idea that I think is the most important one when we're looking at muscles to decipher or decode activity in the central nervous system, which is the fact that because we take advantage of how these signals get transmitted to different muscles, we kind of have a boost in our spatial resolution of this characterization of neural processes. We can use this kind of activity, I will skip this once and go directly to this. We can combine that to different types of paradigms. So for example, here what we did was to use transcranial magnetic stimulation to very selectively activate specific areas of our brains in a specific region, for example, in motor cortex areas associated to the contraction of the index finger. And we wanted to see whether we could find any evidence of changes in activity happening at the brain level that could be characterized based on muscle activity and see how that related to ongoing activity in the brain using standard methods like electroencephalography. So what I show you here is the result of delivering transcranial magnetic stimulation pulses over M1 of the area that projects to the FDI muscle, the finger first dorsal inferior sus muscle, while subjects are producing a sustained contraction with their index finger. And what you can see here, which is pretty much in line with the literature, is the evoked activity in terms of spectral contents that we can produce when delivering single pulses. These are all pulses that are delivered at 90% of active motor threshold while subjects are producing an active contraction, which means that we are delivering stimuli sub-motor threshold. So these are not high enough impulses to produce motor evoked potentials, but instead they are just below that. And we can see here that these kind of pulses produce very widespread changes in terms of spectral contents that we can recognize from the brain. And another thing that you can see here is that this kind of activity, because of the very large effects that transcranial magnetic stimulation has on electroencephalographic signals picked up from the brain, you can see that there's very little spatial resolution in the kind of changes that we observe cortically, right? So most channels are providing us with pretty similar information that is hard to separate or dissociate, despite the fact that we're stimulating somewhere around this area. Okay. Now, if we look at what happens in the muscle, this is the kind of information we have. So these are the responses that we can sense when we look at the first dorsal interossseous muscle, which is the muscle that we are targeting with our stimulation, and then the control muscle, so to speak, which is another muscle that is also being activated is somewhat nearby in terms of representation area. This would be the tibialis anterior. It's not that far away, it's 2 channels away. So it would be somewhere around here, which is also activated, but is supposedly not being targeted by stimulation. So what you can see here, very much in line with this figure that I showed you before, is that we can see 2 things that are very important here. First, we have a massive improvement in terms of spatial resolution. By looking at muscles and how they sample this kind of ongoing cortical activity that is streamed down, we can see that only in those muscles that are specifically connected, mechanically connected with the areas that are being targeted by our stimulation, we can see actual effective changes in the muscle activity. While other muscles nearby, despite the fact that cortically we don't see any evidence of actual changes in this kind of activity in these areas, we see that there's nothing going on. What does that effectively mean? That effectively means that the neurons that are connecting these circuit areas with the muscles are not being entrained by this activity. So it's a way to kind of isolate the activity and the behavior of specific neuronal cells that are task-related or functionally related. The other thing that I think is very important in this kind of results is the fact that, as we can see, and this is important as well, only specific frequency bandwidths are being transmitted reliably. In this case, it's something that goes between 13, 14 hertz and 30 hertz. If we run cluster-based permutation analysis to see for statistical changes, we can see that a broadband of activity is actually being transmitted and producing changes at the muscle level. But we can see that there is a very clear gain or amplification of these beta components at the muscle level, which is very much not related to what the initial components of the TMS evoked cortical activity represents. So it's another thing to keep in mind, which relates to the fact that maybe not all the frequencies are equally well transmitted from the brain to the muscles, and we can maybe think about the fact that maybe not all kinds of activities that are maybe entraining and driving activity of this circuit connecting brain and muscles are going to be equally decomposable in our framework, okay? Yeah, so this is just to wrap up. We have covered this kind of idea of using muscles in order to estimate or decode specific aspects of ongoing activity in the brain. And so long we can kind of say that under ideal conditions where the kind of activity that drives muscles are continuous processes, we should be able to characterize very neatly what are the neural activities that are driving population, and that way we could trace back what this activity looks like and how it relates to brain activity. However, there are some certain limitations or things that we have to consider and keep investigating on in order to further validate this concept, among which I think these 2 ideas that I mentioned about the kind of inputs or the characteristics of the inputs at the motor neuron pool, how do they look like actually, and how we can actually separate the different types of processes, and then what are the characteristics of this information transmission between the supraspinal circuitry and the motor neuron pools. And with that, I think I wrap up. These are some collaborators that we are currently working with, and I thank you for your attention.